Well, we waited enough, so let's start. Okay, so we have a question from Balnur, and the question is, does glucose converted to glycogen by glucagon? No, it's not like that, because glucagon is actually making everything vice versa. So it makes glucose from glycogen and proteins, which means it's the antagonist of the process that you're talking about. So, and muscles, they regulate the consumption of glycogen themselves, so they are not bound to the systemic regulation of the glycogen in liver. Insulin makes the glucose enter the cell, and after it enters the cell and cell senses a lot of energetic molecules, it also either transform it into glycogen or into triglycerides. Whenever we don't use this energy and still consume sugars, they will be converted into long-term energy storage, which are the fatty acids and triglycerides. Do lipids store energy forever? If our body does not use long-term energy, then do our fats just lose energy? No, the storage is quite constant. So you might hear from other people that it's really hard to make these fats go, because they do a lot of exercises to just get a little slimmer. And it means that adipose tissue is really efficient in storing. So to really make these lipids go, many people need to put a lot of efforts and really do heavy physical exercises. And when our body does not use it, the body senses the need to store more. So if people start gaining weight and do not do the exercises, their body thinks, okay, let's prepare for bedtime. Let's consume more energy and store more energy. So the fat never loses the energy storage. Just remember this. Another question is about the transcription and translation. Okay, so what is transcription? Oops. Okay, so what is transcription? And it is kind of copying the gene that is located on the chromosome. So let's say this is chromosome, and this is some protein gene. It is, of course, DNA. And we need to make a replica of this gene, of this DNA. And we make it through the process called polymerization of RNA. And this polymerization is controlled by the transcript factors, and it really makes the copy of this region. So it starts synthesizing the RNA, which will be processed and then will be called messenger RNA. So this process is called transcription. And if you think about the Latin word, scribus means writing, so trans, writing. It means we just copy something from one place to another. And is just a copy made with RNA nucleotides. Now we go to the process. So this is about the transcription, and we go to the process of translation. Just think that in DNA, we wrote a piece of information in the language of DNA, okay? And now we need to translate it with some translator. Into the language of proteins. So this translator is the ribosome. We have two pieces, one bigger, one smaller. And we give this instruction or this writing called messenger RNA. So we have messenger RNA and the ribosome that reads this information piece by piece. And every small piece contains 3 nucleotides. And these 3 nucleotides, they are called specially codons. And this codon, kind of, for this ribosome, this codon means one amino acid. So it's kind of translating from one language, the language of nucleic acid, into another language of amino acids. And this ribosome, it translates the language of nucleic acids and builds the same or equivalent chain of amino acids according to this messenger RNA. So in the end, we have the polypeptide chain that will be processed afterwards. That's why we have transcription, and this script means write. And translation is converting from one language to another. So just remember, transcription is just copying, so from DNA to RNA. And translation means from RNA to proteins. So the question from Alia is, do formed proteins just control some activities while those with which we digest are different? Actually, we digest more own proteins than we consume with the food, because the lifetime of one protein could be just seconds or even days, but we need to recover amino acids that are the building blocks for these proteins. So the proteins, they not just control some activities. As you can see in the lecture, they have a huge spectrum of functions. And whenever they are outdated and we need to renew those, we just digest them. And usually cells digest them themselves, but sometimes we can do it through the alimentary tract and digest it in a hard way. So the question from Gohar, which factors cause the denaturation of the proteins, pH or temperature, and what else? So everything could do that. So the concentration of electrolytes within the same space, which means in the environment. And of course, the temperature, the pH, what else? Of course, the presence of free radicals, because they can bind to the protein and make one single molecule which binds to this protein, could really change the conformation, so it will not get to the previous state, the state of activity.